I'm Mustafa Sayem from Boston Children's, and it's my pleasure to introduce the next speaker, Jurriaan Peters. One of the gaps in our understanding of autism is our lack of understanding of the relationship between autism and epilepsy. NIH recently organized a symposium just on this topic, and it was clear from attending the symposium that there's a group of scientists that study autism and there's a group of scientists that study epilepsy. But they use different vocabulary, different language, different research design. They use different outcome measures. So we really need a new generation of investigators that can fill this gap, that can bridge this gap. And that's where Jurriaan Peters comes in. He's a child neurologist with us at Boston Children's. He's trained as a clinical epileptologist. And then for his research, he's chosen the area of autism, especially within the tuberous sclerosis complex population. And he's going to tell you about some of the research he's been doing with me and Simon Warfield on connectivity as measured by both MRI imaging as well as EEG studies to date.
Thank you, Mustafa, for the introduction. I'd like to talk to you today about brain functional networks in tuberous sclerosis complex and autism. And one of the approaches I'll be talking about is a graph theoretical approach of EEG connectivity. As Dr. Sung has already explained quite nicely, there has been a lot of buzz about autism being a developmental disconnection syndrome. This model hypothesizes that there is a decreased long-range integration of locally processed information, and that can be combined with an increased local connectivity, meaning that these little functional units are locally overconnected and process information. The failure of integration over these longer range distances can impair the ability to form a coherent concept. And this has been shown not only through anatomical studies, as Dr. Sung was mentioning, but it's actually been quite a consistent finding across multiple modalities, including fMRI, EEG, and DTI studies, which I will talk a little bit about. And it is a model that has also been referred to as the first firm finding in autism, where many people were searching for common mechanisms in autism. It also may prove an opportunity for an early biomarker. So we'd like to apply this model to a genetic disorder, tuberous sclerosis complex. It's been mentioned multiple times today. And the molecular pathways are actually very well known. I may dare to say probably most well described of the monogenetic causes of epilepsy. But the genotype-phenotype correlation is poor. And in fact, the genotype imaging correlation is poor. And you can see these tubers here in the brain. So it's a overgrowth syndrome where cardiac rhabdomyomas and brain tubers and several hematomas in other regions of the body, including the lungs and the eyes, can affect these patients neurologically and otherwise. But the imaging correlations of abnormal neurological outcome, including autism, epilepsy, and intellectual disability, are very poor. Multiple attempts have been made to correlate the tubers, which are a striking phenomenon, to the neurological outcome, but this relation is definitely not consistent. Another advantage is that these patients are diagnosed at birth, often with tuberous sclerosis or even before. They have early seizures, but their cardiac rhabdomyomas may be picked up on fetal ultrasound. And the tubers can be picked up with fetal MRI, so when the baby's actually still in the mother's womb. And that provides the opportunity to monitor these children and actually see in about 50% of the kids with tuberous sclerosis will develop autism. So you have a population that by natural course you can monitor the development of autism or not. And because they have bad epilepsy, often up to 90%, and because they have less benign tubers deeper inside called segas, they also undergo frequent imaging studies and EEG studies, so it provides a wealth of data and opportunity to study. We first applied this to a population of 40 children, and we studied the Corpus Callosum. We chose the Corpus Callosum because it is a major connecting pathway between the two hemispheres, and it represents, in a way, the long-distance pathway that we were talking about. DTI, diffusion tensor imaging, is a diffusion-weighted technique in MRI which looks at the diffusion of water molecules, and you can infer anatomical barriers if water cannot diffuse in a certain direction. If you line up all these abnormal diffusion or these preferential diffusion directions, you can build these beautiful three-dimensional tracts, in this case the corpus callosum. And when we did that on 40 children, we found, with tuberous sclerosis and 29 age-matched controls, we found that the structural integrity of the corpus callosum was affected in tuberous sclerosis, but more so affected in tuberous sclerosis when they were autistic. Just to illustrate, this is a subject of 5.3 years old, a control, a tuberous sclerosis patient but without autism, and a patient with autism and tuberous sclerosis. And of course I picked the worst corpus callosum to show you, but the structural integrity of this corpus callosum is severely affected. However, when we go from structure to function, we could wonder what is the impact of these abnormal structures and abnormal connections on the function of the brain. And when we look at structural connectivity and also at functional connectivity, can we find patterns that are not only specific to tuberous sclerosis but also specific to autism within tuberous sclerosis population? So, going back a little bit more, we showed similar abnormal microstructure of the arcuate fasciculus as a white matter pathway heavily involved in language processing, and also in the geniculocalciferine tract processing visual information, for example, for social cognition. We went from structure to function through EEG. We had 46 controls and 42 patients with tuberous sclerosis. And of these 42, 14 had autism. The interesting part of this structure of this population is that these EEGs were clinically available, so nobody had to be subjected to this test for no particular reason. But we also included 16 patients with autism but without tuberous sclerosis. So therefore we could look specifically at effects attributable to autism or effects attributable to tuberous sclerosis. We were looking at connectivity measures, but similar to large data sets that Dr. Sung was talking about, if you have 19 electrodes on the head in a clinical setting or 128 electrodes in a research lab, you get 19 times 18 divided by 2 is 177 possible connections. If you do 177 possible connections and you have several groups, several frequency bands you can examine, and you get a large number of comparisons and a high false discovery rate. So we needed to have specific questions in mind when we embarked on this project. And we first did a conventional analysis, and then in order to not have to look at too much at local connections and random connections, let's say from P7 to C4 or from P4 to F7, we looked at the network as a whole and see how it behaved. And I will give you a better introduction about network theory a little later. As a measure of connectivity, we chose coherence. It has weaknesses and strengths. The main weakness is the skull connectivity, meaning that the adjacent electrodes often bear a similarity, not because of the underlying source, but through conduction of the skull. These are four exemplary subjects from our study. In the top here, a control, and you can see that there are the electrodes are now spread out over a ring so that we can see the relationships a little bit better rather than projected onto a head. And you can see that there are short-range connections between adjacent electrodes. This is a weak pointer. And then there is also strong connections across. There we go. And so the darkness of the blue lines indicates the strength of the coherence. If it's very dark, it's close to 1. If it's absent, it's 0. Here is a patient with tuberous sclerosis, both with decreased short-range connectivity and decreased long-range connectivity. Here's a patient with autism, actually excitingly fitting well in the model of this developmental disconnection. Increased local connectivity and decreased long range. And here's a patient with tuberous sclerosis and with autism, and he has a mixture of the autistic subject and the tuberous sclerosis subject without autism. There are some connections preserved, but the ones that are preserved are mainly in the short range. So when we do the math on that, we can see that the tuberous sclerosis patients here grouped in red and in orange have a decreased mean coherence. So that's overall any possible connection. When we look at the connections between the hemispheres and compare them to the connections within the hemispheres, we can see that there is no group that stands out. On the right here, there's a gray bar, and that was just for a certain control that I tried to introduce. These are 16 patients with an absent corpus callosum, just to see if this measure would work. We would have expected, in a way, since the corpus callosum is so implicated in both tuberous sclerosis and in autism, that there would be a difference with the controls, but there's not. And then long over short range connectivity as a ratio is significantly decreased in patients with autism, and that is regardless of their underlying disorder. So the idiopathic or those patients without a known medical or genetic cause of autism are here in purple, and then with TSC in red, and they actually contribute equally. So TSC is characterized by an overall reduced connectivity, and autism is characterized by a reduced long over short range connectivity. Now, rather than looking at local connections and doing big group analyses on large electrode sets, we wanted to introduce a way of zooming out and looking how the network would behave as a whole. So the problem was originally phrased in a city called Koningsberg in then Prussia, now Russia, and there is a landmass that is locked in by the river Pregel, which goes around it, and there's a total of 7 bridges. You can make this into this cartoon, and you can see the river go around these land masses. And the mathematical question was, can you walk through the entire city, crossing each bridge only once? And you can quite see, if you start here, you go into the island, off the island, back on, off the island, into this one and that one, and you cannot get everywhere with crossing each bridge only once. And you can make this cartoon into a more abstract concept by making the land masses into nodes and the bridges into the limiting factor called the edges, indicating the connection strengths. The shape of the edges, and in a way the shape of the nodes, is not really relevant because you're working in network space rather than in physical space. But we still have preserved some of the anatomical relations. Now, you can do same things with networks in the biophysical world or in the technological world. For example, airline networks in North America, electrical grids, and in the brain. And there have been beautiful studies in the brain, for example, showing connectivity matrices of DTI connectivity in a growing, in an aging population, showing that there is increased connectivity and certain network properties are very strong, and then later on representing are decreased, representing overconnection and subsequent pruning, thought to be one of the mechanisms in the normal development. So when we wanted to apply that to EEG, we wanted to take a few measures into account. The first, average path length. In a network, what is the average path length when I try to get from A to B, or let's say from A to E? And if you have an isolated network node, the average path length goes up. So for example, if you would compare this with airline networks, if you would have a few isolated airports in, let's say, rural Africa, then the entire African continent network average path length would go up. So it's dominated by isolation of nodes. The clustering coefficient tells you something about the segregation rather than the integration. And it tells you if this node is connected with that node and this node is also connected to that node, what is the likelihood that these two nodes are also connected to each other? So it's a measure of triangulation. You can see that the right side of this graph is more has a higher clustering coefficient than the left side of this graph. And finally, I think one of the most appealing ones, if only by the name, is global efficiency, which is driven as a measure of integration, but which is driven by the strongest path. So in an airline network, that would be the connections between London and Boston, or between New York and Boston, if you will. And this is also the measure that we use a lot when we try to later on attack these networks and subject them to stress. Lastly, small world networks are networks that are both locally very segregated but also integrated. And people know this best by the 6 degrees of separation, through who everyone is connected with Kevin Bacon or something like that. Average path length, clustering coefficient, and global efficiency were again decreased in the tuberous sclerosis population, regardless of their autism. The presence of autism, yes or no. A little bit surprising, because based on the theory that we discussed earlier, you would have thought that maybe there would be an increased local connectivity, higher clustering, or a longer path length, more isolation. But there's not, so the network topology or the network design is not fundamentally altered, at least by this measure in this EEG study with this particular frequency band. So tuberous sclerosis has an increased path length, decreased clustering coefficient, and decreased efficiency. Now, quickly, the last measure that we did is resilience. You can subject these networks to attack and remove certain nodes. You can imagine an airport strike or a terrorist attack at a big airport, or a failure of a power grid, and you want to see which cities are going to be affected depending on how connected these nodes are. And in the brain, it works the same. For example, in Alzheimer's disease, highly connected nodes are, such as the basal ganglia, are in neurodegenerative disease in general thought to be metabolically more active and therefore may degenerate first. So the targeted attack, where the highly connected nodes are taken out first, seems to be biologically a little bit more relevant. So when we do a targeted attack to our EEG networks, we can see that with the removal of every node, the global efficiency goes down from 100% of its original value, goes down stepwise. This is just a cartoon that I made up. But if you look at our populations again, we can now see that the autistic population stands out clearly, and not with decreased resilience, what you may think intuitively, but actually with increased resilience. So the network is unperturbed. So autism is characterized by decreased long-range connectivity, an unaltered network design, and an increased network resilience. And if you put that on a little plot, you can actually separate the groups out again. It fits with the theory of developmental disconnection, and the increased resilience is a little bit harder to explain. But if you think of resilience as an indicator of redundancy of the network connections, for example, if you have suspenders and a belt, and you take away the belt, the pants will still stay up. So if you remove a node and the network does not respond, it may be that there is a certain level of redundancy and that there are other areas that take over the exact same function. You can imagine doing a smile with your basal ganglia or making a smile with your motor cortex. And this concept of redundancy or degeneracy is maybe in place, indicating that these nodes are not as specialized. So functional and structural data are complementary. The fact that these findings of an increased resilience, but also the long versus the short range distance, are specific to autism, regardless of the etiology, tuberous sclerosis or not, puts tuberous sclerosis on the map as a model for autism, and it could be used as a biomarker for clinical trials. And I really think that including heterogeneous populations to find common biological mechanisms is a way to find unique descriptors of autistic properties and that are not just characterizing a subgroup. Thanks very much.